Brad Larson from BioTek Instruments. I'd like to briefly tell you about a project that we did collaboratively with Kuraray, Elplasia to demonstrate the ability to perform imaging-based apoptotic phenotypic assays using the 384 well Elplasia microspace plates. The goal was to place different cell types within the plates and allow for spheroid formation and then track apoptotic induction over time using live cell imaging. We used the Cytation 5 cell imaging microplate reader to perform all the imaging that was carried out, and then the Gen 5 cellular analysis software to analyze the data from each image. Here we're seeing first the ability to image the spheroids using a bright field. This is a single well of the 384 well plate using a 4x objective and a 2x2 montage. We then demonstrated the ability to also use fluorescence imaging. Here you see the same well with the spheroids that were stained with a Hertz 33342 dye to identify nuclei within the spheroids and allow us to see each spheroid in the well. We then induced apoptosis with different compounds and tracked apoptotic induction with a reagent that was able to bind to externally exposed phosphatidylserine on the surface of the cells. So therefore, increased green fluorescence is an indicator of increased apoptotic induction. We then used a number of parameters in Gen 5 to accurately identify the spheroids, and then with subpopulation identify only or analyze only the spheroids that we're interested in, and also determine the number of apoptotic spheroids within each well. That allowed for object mask placement around all spheroids, as you can see on the left, and then subpopulation to, again, only analyze the spheroids that we were interested in. And then by using the fluorescence from the green channel from the apoptotic reagent, we were able to identify a subpopulation of apoptotic spheroids within the well. That allowed for us to track the number of apoptotic spheroids in each well with using different cell models and concentrations of drugs that were tested. And then by dividing by the total number of analyzed spheroids, we can normalize the data to get a much more accurate and robust analysis. We can also look at a single time point with different concentrations of drugs, as you can see here. So you can either perform endpoint analysis or, again, live analysis using kinetic data. If you'd like further information, you can go to this website to see more about how the project was performed. And in the end, we feel that this is a very robust, high throughput, high content way to accurately measure phenotypic apoptotic data using the Elplasia 384 well microspace plates. Thank you.